Come on. Okay, are there any questions? I'm not hearing anything, so let's begin. Boy, that's not right. I scroll that down a little. Let me share my screen. Can somebody confirm you can hear me?
I can hear you.
Thank you. All right, you should be seeing my screen now. Today is March 6th, and we are supposed to be starting chapter 13. We're not there yet. Hopefully, we'll get started before the end of the day. But we're on chapter 14. Let me remind you, you do have a quiz you need to take by 11:59 p.m. tonight. It'll cover chapter 8 and lab 9. I did get lab 9 graded. I think that was late last night when I finally got it graded, although your grade for the other than the case studies had been graded not last night, but Monday night. There is no lab today. I will be logging in at the normal time for 15 minutes to answer any questions. Do note that your unknown notes page is closed Friday, March 8th. That's this Friday. And the unknown report is due this Saturday. Any questions about any of that? The final will be Wednesday, March 20th. You must complete it by 11:59 p.m. It'll have heavy coverage of chapter 7, 13, 14, and lab module 9. Excuse me, I guess that's lab module 8. I'll look into that. um, Any questions about any of that? We will have a review during the lab next week. It'll probably be on Thursday, where it'll be a review for the cumulative final. It is cumulative and comprehensive, but it'll be heavy coverage of the chapters you have not been quizzed on, meaning chapter 7, 13, 14. And I'll look into the lab. I don't know if it's lab 9 or lab 8 that has the heavy coverage, because I've got lab 9 on this quiz. All right. If no questions, let's begin. Seems like there's one more thing I wanted to mention. Up to me.
Can I actually ask a question real quick about the unknowns project?
Yes.
So, the notes are the notes because we're not actually running the test. Is the report, I mean, vastly different from kind of just the notes we were doing online? I mean, I'll have pictures and things like that included in it, but
You can take a look at the sample journal. It gives a report. So it's an example of a report. If you write your lab notes very formally, meaning the unknown notes pages very formally, there won't be a whole lot of difference between the report and your unknown notes, other than that the report has some topics that must be in there, and you don't need those topics in the unknown notes.
okay.
Okay.
All right, thank you.
All right. So I have that we discussed the severity and duration of the disease, and that we had talked about this slide here, talking about acute diseases and chronic diseases. Let's talk a little bit more about the extent of host involvement in a disease. Infections can also be classified according to the extent to which the host body is affected. A local infection is where the pathogen is limited to a small area of the body, such as a boil of the skin. Being a skin infection, like if a Staphylococcus aureus or maybe MRSA, not methicillin, multi-drug resistant Staphylococcus aureus, causing the skin boil. An abscess is also a local infection. A local infection differs from a systemic or generalized infection, as this infection is one that spreads throughout the body. Like when a patient gets the measles, that becomes a systemic infection where you get it on your skin, you get it in your blood, you get it essentially everywhere in your body. A focal infection is a systemic infection that began as a local infection, such as MRSA, starting from a skin boil. If it invades the blood can then spread it throughout the body. Why it's called a focal infection that develops into a systemic infection. Any question about any of that? Bacteremia is the presence of bacteria in the blood. When clinicians monitor their blood, they will usually look for bacteremia. Septicemia is something similar, but this is actually growth of bacteria in the blood. Most clinicians cannot determine septicemia. You'd have to send the blood to a lab to determine septicemia, at least in most cases. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. And there's a picture of streptococcus. That might be streptobacillus growing in the blood. It's probably streptococcus, though. Not growing, but present in the blood. We don't know until you do a test to see if the bacteria is growing in the blood that you can call it septicemia. The host of resistance also determines the extent of an infection. A primary infection is the acute infection that causes the patient's initial illness. A secondary infection is usually an opportunistic infection after a primary infection predisposes the patient to getting the secondary infection. Usually, the primary infection weakens the host, allowing the secondary infection to occur. I'm trying to think of an example of that, like you can get a respiratory disease causing damage to your mucous membrane, and then you can get a bacterial infection growing in the weakened mucous membrane, where the mucous membrane is no longer entirely intact, and then the nosocomial infection can become, not a nosocomial infection, sorry, the normal microbiota can then become an opportunistic infection. One example might be, it's frequently seen in children. They come down with a cold and their ears get stuffed, and then they get an earache where bacteria start growing in the ear. A subclinical disease is a disease where the patient has no noticeable signs or symptoms. We've called that an asymptomatic disease before. So asymptomatic, subclinical, same thing. The patient has no signs or symptoms, but they do have the disease-carrying organism, and they can spread the disease to others. I've mentioned that COVID-19, young adults, children, frequently have a subclinical infection where they do not know that they have a COVID-19 infection, and yet it's growing in their body, not causing them any trouble, but then they can go and spread it to somebody else like their grandparents. And about 50% of young people have that with COVID-19. It's known that some people can carry poliovirus, never having signs or symptoms of polio. Let's continue talking about the severity or duration of disease, two terms we need to know. And talking about the severity or duration of a disease are a latent disease and herd immunity. A latent disease is a disease where the disease goes away, but the disease causing organism goes latent, meaning it doesn't go away. It just goes sort of like into hibernation. And then later, the disease causing organism can come out of hibernation in the latency and then re-cause disease. The best known example of that is a child can get chickenpox and then the virus goes latent, and it can go latent for decades. Usually it will come out not when the child is still a child, but when the child is an elderly adult, and the virus will come active and cause shingles, causing disease again. When the virus goes latent, it is generally just sitting out, not causing any diseases, not being metabolically active, and just waiting for the right time to come out. Usually, there is a stressor which causes the virus to become active. And in the case of chickenpox, the stressor is usually old age, where the patient's immune system weakens. And then the virus can come out and cause shingles. All other times the virus comes out, it'll cause shingles. The first time it causes chickenpox. Another case of a latent disease is the cold sore. When you have an active cold sore, you have the disease. And then the virus goes latent, hides out, and waits for a stressor, and then the virus can reactivate, usually a year later, causing the cold sore. And you can tell from the name of the cold sore what the stressor usually is. When a patient gets a cold, they can get the cold sore. Any question about any of that? Okay, herd immunity is when many people in the population are immune to the disease-causing organism, so the disease cannot spread. Let me blow this up. With the population not being immune to the disease-causing organism, when the first people become infected, the disease will then spread to the non-immune people, and it'll just continue spreading and continue spreading until the majority of the people become infected. And actually, this was what they feared would happen with COVID-19, because the world had never seen it before. Nobody had immunity to it. And then it spread pretty easily. Actually, this is why the flu epidemics happen. A new flu virus comes around that people don't have immunity to. And then somebody gets the flu and it just spreads and spreads. Although the flu isn't as contagious as COVID-19, but that's why the epidemics happen with the flu about every 10 years, because a new variant comes along and the population doesn't have any immunity to the new variant. Now, obviously, if you get the flu shot and they did their job and did a good job of anticipating what virus is going to be coming down the line 6 months, the flu shot should protect you. But every once in a while, they miss what will be the flu variant 6 months down the road. And occasionally, because usually the flu shot protects like 3 or 4 different viruses, occasionally they've missed on all 3 of them or all 4 of them. Not often, but occasionally they do. In which case the flu shot doesn't give you much immunity to the flu spreading that year. It's been a long time since that happened, but I remember when that happened. I think it was in early 2000 when all 3 of the flu strains spreading were not in the flu vaccine. Now, with herd immunity, the majority of the population are immune. They're the yellow people here. And then when somebody becomes sick, only the people in close association with that sick individual will become sick. And then the other people who are not immune are protected because the disease causing organism has to go here or here before it can go there. And this individual is immune, so is that individual. So this individual is never exposed to the infective agent. So herd immunity protects the herd from the disease spreading. And what it does is it actually protects the non-immunized people. Now, with some diseases like measles, you need 95% of the population to be immune before you get herd immunity, and then the disease will not spread. Every once in a while in the United States, we have measles spreading, and that's because the number of children vaccinated is not 95% or higher. With measles, it actually is pretty high. It's around 90% of children are vaccinated, but that's not high enough to keep measles from spreading. And so every once in a while, measles does spread in the United States, and it's mostly from the parents who say, oh, measles isn't a problem anymore. I'm an anti-vaxxer. I don't want to vaccinate my children. And then there are the children who are most likely to get the measles when it spreads. It actually frequently spreads in places like, oh, what is that called? The Mickey Mouse, Walt Disney World, or Walt Disney, whatever the other place is called, Walt Disney World, where a bunch of children are all together. You only need one child to be sick, and then all the non-immunized children are exposed to it. Any question about latent disease or herd immunity? Let me briefly mention that COVID-19 needs at least 85% of the population to be immune to keep COVID-19 from spreading. In the United States, that'll never happen. The highest percentage we ever got was when the vaccine first came out, and that vaccine no longer prevents disease. It no longer gives immunity to the current strains of COVID-19. And like I said, it was around 70%. And nowadays, the people getting vaccinated are much less than that. I think I read the latest vaccine of COVID-19, only about 30% of the population got that vaccine. I was one of those 30%, by the way. And until the population becomes 85% immune to the disease, COVID-19 will continue spreading. And with the vaccine, that'll never happen in the United States. People just aren't going to get the vaccine. And it is true that some people can have COVID-19 and then have immunity, so that the immune people are more than just the vaccinated people. The trouble with COVID-19 is when it hits a person, the immunity it gives that person is not long lasting. 6 months to a year at most, and then the immunity wanes. Any question about any of that? All right, let's talk about patterns of disease. I can't get that out of there, so let me move that over there. Normally, a specific sequence of events is associated with an infection by a microbe, and then the subsequent signs and symptoms of disease in a patient. Excuse me here. Hopefully you didn't get to see any of that. I tried to get out of the window. Before disease happens, the disease-causing organism must come from a reservoir. A reservoir is where the disease-causing organism survives, where it exists before it infects the patient. The pathogen must be transmitted to a susceptible host. The pathogen has to invade the host and multiply in the host. And furthermore, the pathogen has to injure the host. If you do not have injury, you have no signs or symptoms of disease. The illness will then end when one of two or three things happen. First, the host immune system destroys the pathogen, and then the illness ends. Second, the pathogen can kill the host, and then the illness ends. And there is a third option, and it is when the pathogen just never goes away. When HIV infects a patient under modern medical care, HIV never goes away. The illness may never fully end, although with modern medication with HIV, you can usually prevent AIDS, or once AIDS establishes, you can get the patient's health to get better and then AIDS goes away. But they're always hanging sort of like a, what is that? What's the expression? They're always have like a knife to their throat, or a gun to their head, whatever the expression is. And that is HIV is always there and it can always come back. And it is true that HIV patients now live almost to a normal lifespan, but it is a little shorter. They live about 5 to 10 years less than a normal healthy individual. And that's with modern medication, where the patient is getting first world medication. All right, any question about any of that? Certain factors can predispose a host to making them more susceptible to, and therefore affect the occurrence of disease. For example, females have a shorter urethra. Therefore, they have a higher incidence of urinary tract infection and bladder infections than males. It's just the longer urethra of males protects the bladder. Inherited traits of sickle cell anemia gene make the patient more susceptible to getting disease. Another inherited, well, it's not inherited trait, another trait is diabetics are more susceptible to getting a disease. Diabetes is not an inherited trait, it's a multi-complex relationship. So there is some genetics involved, but there's a lot of environment involved in diabetics. The point is, a diabetic is much more likely to come down with any other disease, especially an infectious disease, than a normal healthy individual. Climate and weather is known to be predisposing factors. Everybody knows the colds and flus are more likely to occur in the wintertime or the late fall and early spring. There's a couple of reasons for that. We won't talk about the reasons, but the point is climate and weather can predispose you to getting disease. Fatigue can also predispose you to getting disease. When you're fatigued, your immune system is weakened and your body's defenses are weakened, making you more likely to coming down with an infection. Let me warn you that students are frequently fatigued, either from too much studying, too much work, or too much partying. And when you're fatigued, you're more likely to come down with an infection. Age is well known to cause disease. Let me see if I can get this. I don't know why that happens. Sometimes when I click on here, I don't get paint to come. This is not even the right thing to come. I want my computer to come, not that. This is Color College. I don't know. Oh, there it is. Never mind. Paint is there. So normally, when we look at the age and the number of cases, so the number of cases on the y-axis, the age on the x-axis, most diseases follow this distribution of a disease, where the very young are more likely to get disease, more likely to get serious disease, and the very elderly are more likely to get disease, more likely to get severe disease. That's true for almost all, well, most diseases. We'll say most diseases. It is not true for COVID-19, where only the very young have an elevated risk of COVID-19. And then the elderly have quite an elevated risk for COVID-19. So the newborns are at risk for COVID-19. And I'm not sure where that drops down to very low levels, but I know it's like age 3, COVID-19 drops very low. And even at age 1, COVID-19, it's probably pretty low. I don't know where it is, but let's say that's at age 1. I don't know. But my point is it drops significantly from newborn to age 1. So very rapidly in children, the risk of COVID-19 drops. And then in, I don't know what the age is, but if at least an elderly child, I'm not talking about a teenager, I'm talking about something like 10 or 9, the risk of COVID-19 is very low. And that is very low for teenagers, young adults, and it doesn't start climbing until, you get starting approach middle age, and it doesn't become high, and I don't know where you call high, it doesn't really become high until about age 65. But it does climb steeply in the elderly, and then it keeps climbing high, meaning the older you are, the greater the risk of COVID-19. So COVID-19 mainly hits the elderly, but it does hit the very, very young, the newborns. Any question about any of that? Lifestyle can affect your chance of getting a disease. It's well known that smokers, for example, are more likely to get a respiratory infection. The reason is smoking damages the mucous membranes in the upper respiratory tract, and that makes the patient more likely to getting a respiratory disease. Chemotherapy is another predisposing factor, mainly because chemotherapy weakens the immune system, and without having a good, strong immune system, you're much more likely to come down with an infection, and it's much more likely to be a severe infection also. Any question about any of that? All right. When we're talking about disease, you should realize that disease normally goes through certain stages of development. Once the host defenses are breached, the disease follows a certain sequence of stages. I'll word it that way. The sequence tends to be similar for both acute and chronic diseases. So most diseases follow this development of disease. First, there is the incubation period. That is when the patient first becomes infected with the disease-causing organism till the time when the patient first has signs or symptoms. When the patient first has signs or symptoms, the incubation period is over. So in the incubation, the disease-causing organism gets established in the patient and then starts multiplying and obviously starting to do damage to the patient. But the patient doesn't have any signs or symptoms, and that's the incubation period. The prodromal period follows, and that is when the patient first has mild signs and symptoms. So like when you're walking down the street and you say, geez, do I have a sore throat? And you have to think about it. And you say, yeah, I've got a mild sore throat. I must be coming down with something. That's the prodromal period, where the signs and symptoms are mild. The period of illness is when the signs and symptoms are no longer mild. This is when the signs and symptoms reach their greatest and worst extent. In the period of illness, besides being the signs and symptoms over here, meaning signs and symptoms worse up here, it's also the number of microbes. The number of microbes are highest in the period of illness. So they start very low in the incubation period, and then the number of microbes increase in number in the patient, and they're highest in the period of illness. The period of illness is followed by the period of decline. This is when the patient starts to feel better. For example, when a patient has a fever, the period of decline starts when the fever breaks. Any question about any of that? The number of disease-causing organisms decreases in the period of decline. They decrease until they reach a threshold number. Oh, excuse me here. Until they reach a threshold number where the number of microorganisms are low enough that the patient has no longer any signs or symptoms of disease. The period of decline then ends, and we call that the period of convalescence, when the patient still has the disease-causing organisms, but they no longer have any signs or symptoms of disease. Any question about any of that? All right, here's a question for you. In which period of illness is the patient no longer communicable? The patient is not capable of spreading the disease.
In the period of decline or
Period of
convalescence?
decline. That's a good guess. Anyone have another guess? Meaning, sorry, that's wrong.
Period of convalescence.
Period of convalescence. That's a logical guess because you no longer have any signs or symptoms. And most people would think, oh, I don't have the disease anymore. But actually, in the period of convalescence, you still have the disease causing organisms. You still are communicable, less so than the period of decline, less so than the period of illness. But you still are communicable because you still have the organisms. And I didn't mention it. After the period of convalescence, you don't have the disease, okay? And they don't have a name for that because it's not a period of disease because there is no longer any disease, okay? So it's after the period of convalescence that the patient becomes healthy, and it's after the period of convalescence that the patient can no longer spread disease. The patient can spread the disease everywhere in all the different stages of disease, including the period of convalescence, when you don't even know you have the disease-causing organism in you anymore because you don't have any signs or symptoms. My mother used to always tell me, oh, I'm getting better now. I'm not contagious. And I always told her, Mother, you're talking to a microbiologist. I know better. As long as you have the disease-carrying organism in you, you are communicable. There is slightly one exception, and that is very early in the incubation period. You are not contagious because you just don't have enough organisms in you and they're actively growing in you. And so you're not communicable shortly after incubation period starts. I'm not sure how long that would be, but let's say at least a couple of hours after you're first infected, you're not communicable. But during the incubation period, you are communicable. In fact, for common illnesses like the colds and the flu, for adults, you are most contagious late in the incubation period or in the prodromal period. Even though you have more disease-causing organisms in the period of illness, in an adult, the immune system is reacting to the illness and preventing the spread of the pathogen for common illnesses like the colds and the flus. I think this is because back in our caveman ancestry, parents, there was selection for parents not to spread the disease to their children. And so our immune system ramps up during the period of illness and then makes the patient less communicable than in the period of prodromal period or the period of incubation, at least late in the period of incubation. That's actually not true for children. They are they are communicable mostly in the period of illness. And when I was a child, hospitals didn't allow children into the hospital because they frequently had disease and then they were communicable during all the periods of illness. I know this because when I was 13, I wasn't allowed to go visit my brother in the hospital. I said, sorry, you're under age 13. We don't allow patients like that into the hospital. Most hospitals have changed that because they decided that it is socially advantaged to have like children go visit their grandparents in the hospital. And so they've opened the doors to children. But when I was in the hospital, when I was a child, most hospitals did not allow children into the hospital. All right, any questions about any of that? Let me state that not all diseases have every period of illness. For example, one moment I'm walking around fine and I'm in the incubation period for the flu, and then the next moment it feels like somebody has hit me over the head with a sledgehammer. There was no prodromal period. Sometimes the flu occurs that way, where you go immediately from the incubation period to the period of illness. And then for some diseases, there is no period of decline or period of convalescence. For example, if the disease kills off the patient, there is no period of decline or period of convalescence. In the original days of HIV, I don't know when the modern medicine happened, but let's say from 1980 to 1990s, when an AIDS patient got AIDS, their life expectancy was only about 2 years. HIV and AIDS killed them off, and then obviously they never entered a period of decline or convalescence. Any question about any of that? All right, so those are the different stages that disease can go through. Let's move and talk about the spread of the disease. I've already talked about reservoirs, the source of infection. Reservoirs are the continual sources of the disease-causing organisms. Reservoirs can be living or non-living. For human diseases, humans are a major reservoir. Some diseases like AIDS, gonorrhea, humans are the only reservoir. So the disease-causing organism has to come from a human, assuming that, like I said, HIV or gonorrhea. With many diseases, the carriers may have inapparent or subclinical infections, meaning they do not know they are infected, and they can spread disease. A patient can also have a latent disease, and then it comes out of latency, and then they can spread disease. I can talk a little bit more about that with chickenpox. Like I said, usually a child gets chickenpox, and then the virus reactivates, usually when the child is an elderly adult, like a grandparent. And this virus could then spread from the grandparent to their grandchildren. And that's the way the chickenpox virus established itself. So it hides out, and then it infects the grandchildren from the grandparent who has shingles. And that's just the way the virus has evolved. It's a human virus, and it's well evolved to living in humans. For example, if it does hit children, it usually doesn't cause severe illness. And in shingles, it doesn't usually kill the patient, it just causes extreme pain. And then when the shingles is there, the patient can spread it to other individuals. The point is the virus was in hibernation, latency, for decades. Animals can be reservoirs of disease. Some diseases, like rabies and Lyme disease, humans only get from an animal. We get rabies from the bite of an animal, usually a dog or skunk or fox or something like that, a cat. Lyme disease from the bite of a tick. These two diseases do not spread from people to people, at least not directly. Obviously, a tick could bite a person, pick up the Lyme disease, and then go bite another person, but that's not spreading person to person. Some diseases called zoonoses are animal diseases that can be transmitted to humans. Actually, rabies is a zoonoses. It spreads from animal to animal, but it can spread to humans. The thing about rabies is once it infects a human, the rabies virus does not spread. It's a dead end for the virus to infect a human. It kills the human, but humans do not spread the rabies virus. Any question about any of that? And then, of course, many diseases have non-living reservoirs. An example are botulism and tetanus. They have non-living reservoirs, usually the soil. Botulism could be like canned food. The soil, water, and lettuce are often non-living reservoirs of disease-causing organism, largely causing gastroenteritis in humans. Any question about any of that? If not, let's talk about the transmission of disease. Disease has three principal routes by which the causative agent of disease can be transmitted from the reservoir to a susceptible host. So the disease can spread from the reservoir by contact transmission, by vehicle transmission, and vector transmission. Contact transmission is from patient to patient. Vehicle transmission is by some object transmitting the disease-carrying organism to a susceptible host. That object, we have a name for it. It's called the fomite. Vector transmission is some animal that spreads the disease-causing organism to the human. Any question about any of these three principal routes of transmission? All right, let's talk a little bit more about contact transmission. There are three types of contact transmission: direct person-to-person transmission, requiring close association between an infected individual and a susceptible host. Examples of direct contact transmission are spreading the disease during a handshake or spreading the disease during a kiss. Any question about any of that? Indirect contact transmission is spreading the reservoir to a susceptible host via a non-living object that we call a fomite. For example, somebody's got a cold, they sneeze on their hands, they then touch money, transferring the virus to the money. They then turn in the money to the cash register. The cashier then turns the money over to the next customer. The customer handles the money. They pick up the disease-carrying organism on their hands. So money is actually a good way of spreading disease around. Contaminated tissue is well known to spread disease. And in baseball, the baseball could actually spread the disease-causing organism among the players. Remember, the object is called the fomite, and it's just spreading the disease-carrying organism from one patient to the next susceptible host. The third contact transmission is droplet transmission, and I'm not going to quiz you on that. I'm going to discuss it briefly, but I'm not going to quiz you on this because droplet transmission is contact transmission, and that's when somebody sneezes or coughs and the airborne droplets travel less than 1 meter. So in this sneeze, about here would be 1 meter, and that's considered droplet transmission. However, in that same sneeze, the droplets can go beyond 1 meter, and beyond 1 meter is not considered droplet transmission or contact transmission. It is called vehicle transmission. Let me show you that there. And it's airborne transmission of vehicle transmission. My reason for not testing you on this is I don't like the distinction that 1 meter is droplet, beyond 1 meter is airborne. Droplet is contact, airborne is not contact, but vehicle transmission. Because it's the same sneeze. Doesn't matter where you get it, here or there. If you pick up the disease-carrying organism, you'll get the disease. It's true you get more disease-carrying organisms here than beyond here, but still you can pick it up from the same sneeze. Any question about any of that? All right, showing you an example of direct contact transmission, high-fiving, spreading the disease from one hand to another. Indirect transmission, where the disease-carrying organism is put on the baseball, and then the catcher will handle the baseball with their hand, picking up the disease-carrying organism on their hand. Usually, the catcher would then get the disease when he touches a mucous membrane with his hands, such as the eyes, the nose, the mouth, even possibly the ear, but the ear is less likely. Any question about contact transmission? All right, if not, let's talk about vehicle transmission, where some object tends to spread the disease-carrying organism to the patient. Vehicle transmission is by an inanimate reservoir, such as an object, food, water, body fluids. There are 3 types of vehicle transmission: waterborne transmission, foodborne transmission, and airborne transmission. Waterborne transmission would be something like your water supply, like your well, becomes contaminated by sewage, and then people who drink that water would get infected. This doesn't happen very often in first world countries, but in third world countries, it happens way too often. Foodborne transmission, where the food is either undercooked, poorly stored, or unsanitarily handled. The disease-causing organism gets in the food, and then the food is consumed by the patient, and then the patient gets infected. Airborne transmission is when airborne droplets travel more than 1 meter from the reservoir, in this case, the person sneezing. So out here, airborne transmission. Any question about any of that? Like I said, I won't quiz you on airborne transmission. Let's talk a little bit about vector transmission. A vector is an animal that carries the disease-causing organism to the patient, who then gets the disease. The most important group of vectors are the biting arthropods, especially fleas, ticks, and mosquitoes. There are two ways that vectors can spread disease. There's mechanical transmission and biological transmission. Mechanical transmission is passive, like this fly here is picking up the disease-carrying organism on the feet of the fly, and then the fly lands on the food, passively transmitting the disease-carrying organism to the food. And then the food is eaten by the patient, and then the patient becomes sick. So that's mechanical transmission of disease. Vectors can also transmit disease by biological transmission. In biological transmission, the pathogen is reproducing inside the animal vector. An example of biological transmission is malaria. Malaria grows within the mosquito, and then when the mosquito bites the person, the mosquito can transmit the malaria disease or causing organism, Plasmodium species, into the patient, giving the patient malaria. Biological transmission, where the pathogen is reproducing inside the vector as well as the patient. It has to reproduce inside the patient or you don't have disease. Any question about any of that? If not, let's talk about nosocomial infections. These are also called healthcare acquired infections. This is one of the most important topics I can teach you, assuming you go into the allied healthcare profession. Nosocomial infections, or hospital-acquired infections, are infections the patient gets as a result of a hospital stay or a hospital-like environment, like a nursing home. The infection was not present before the patient checked themselves into the hospital or hospital-like facility. The patient got this nosocomial infection from staying in the hospital-like setting. It's estimated that 5 to 15% of all hospital patients in the United States come down with a nosocomial infection each year. That's 2 million people per year in the United States, or 20,000 deaths each year in the United States from a nosocomial infection. It used to be the 8th leading cause of death in the United States. Because of COVID-19, it may be the 9th leading cause now. So it's either the 8th or the 9th leading cause now. By the CDC, about 1 in 10 hospital patients get a nosocomial infection. And then about 50% of nosocomial infections are preventable. The patient did not need to get it if everyone had taken steps to prevent the nosocomial infection. I've got this information that nosocomial infections are an economic burden on the United States for year 2013. It's estimated it costs $9.8 billion US dollars. You don't need to know any of this, but I'm just laying it out for you. It is a economic burden on the United States. Nosocomial infections result from the interaction of three factors happening in the hospital-like setting. First, microorganisms are present in the hospital environment or the hospital-like setting. Hospitals are major reservoirs of sick patients. These sick patients have organisms, microbes, that cause their illness, and those microbes can spread. Hospitals are also major reservoirs of other microbes, such as human normal microbiota, which can cause an opportunistic infection. Some nosocomial pathogens are also antibiotic resistant, and then this causes a problem because when the patient gets the nosocomial infection, the clinician prescribes the usual antibiotic and it doesn't work. The clinician then orders another antibiotic and usually orders a test to determine which antibiotic to use. But remember, the patient is in the hospital for a reason. By this time, the patient could die from the nosocomial infection before the test ever comes back on which antibiotic to use. Any question about any of that? Oh, we could also say that antibiotics are very prevalent in the hospital or hospital-like setting, so that the frequency of antibiotic resistance is actually higher in this environment. Meaning microbes are more likely to be antibiotic resistant in the hospital than elsewhere on the planet Earth. Nosocomial infections also happen because the patient is in the hospital for a reason. They are a compromised host, ones whose resistance to infection may be impaired by disease, by therapy, or by burns. Broken skin or mucous membrane of the patient can compromise the host. Burns, injections, ventilators, urinary catheters, the administration of anesthesia can all weaken the host and make a nosocomial infection more likely to happen. Furthermore, the host may have their immune system suppressed, such as suppressed by drugs, by radiation therapy, or burns. If their immune system is suppressed, they're more likely to become infected, and the disease will be more likely to be severe. So the patient is compromised. That's why they're in the hospital. They're in their hospital for a reason, and usually they're sick, and so they'll be compromised if nothing else. Their immune system is tired of trying to fight off the disease. And so you give them a nosocomial infection, the immune system will be already weakened. There's also a chain of transmission in the hospital-like setting. The principal routes of transmission are direct contact transmission, such as patient to patient. In special care units, the hospital staff tries to prevent patient to patient transmission of disease, like in the ICU units. If the patient is there for a communicable disease, they try to prevent the spread of that disease. But the hospital staff can also spread the disease. So when they touch the patient, if they're not wearing gloves, they should always wash their hands before going on to the next patient. And if you have gloves on, you should discard the gloves before moving on to the next patient. And it's actually a good idea to wash your hands after discarding the gloves, just to be on the safe side. There's also indirect contact transmission. This happens via fomites, urinary and IV catheters. Contaminated surgical dressing can cause indirect contact transmission. A patient who's in the hospital, and let's say they've got a roommate, the patient turns on the light, they give the disease-causing organism to the light switch, and then the next their patient, their roommate, then turns off the light. They pick up the disease-carrying organism. Any question about any of that? Now that's if a patient has a roommate. If they don't have a roommate, the patient could still spread the disease, like when they go in the elevator going to the cafeteria or something like that, they can spread it to the buttons of the elevator. There's also vehicle or airborne transmission, where the ventilation system can spread the disease. I remember when there was an Ebola patient in the United States. It's when somebody who was treating Ebola patients in Africa came back to the United States because they got infected with Ebola. And the hospitals were worried, geez, this might spread in the air. And their solution was to have the air in that room localized so it didn't feed back into the ventilation system. Any question about any of that? All right, how do we control nosocomial infections? There are certain procedures generally implemented to control the spread of nosocomial infections. First, in microbiology, you've learned how to use aseptic technique. When you go into the health care facility, make sure you always use aseptic technique, and you'll be less likely to spread a nosocomial infection. You should use proper handling of contaminated materials. And if you get the bacteria or organism on your hands, after you're done, you should go wash your hands immediately. Because if you touch something, you could spread the disease-carrying organism to some object, some fomite. Something that's been shown to reduce nosocomial infections by about 50% is simply frequent and thorough hand washing. You should always wash your hands when going from one patient to another so that you are not spreading a nosocomial infection. Then they have educated the staff, the patients, and the visitors to prevent the spread of nosocomial infections. Hospitals in the United States have gotten very good about that. In the 1960s and 50s, they didn't even educate the doctors. If you take a look at the old medical movies, you'll see the doctor proudly wore his stethoscope on his chest, and he would take a stethoscope, listen to the patient's heart by putting it against their skin, and then drop the stethoscope thumping on his chest. And then what would he do? He'd then move to the next patient and use the stethoscope on the patient's skin to listen to their heart. Doctors in the 1950s and 60s were spreading nosocomial infections with their stethoscope. Nowadays, they know after they touch a patient, their skin, with the stethoscope has to be sterilized before it is used on the next patient. My point is, doctors first became educated, like about the 1970s. Then a patient started becoming educated. They started trying to educate the patients so they weren't spreading nosocomial infection. And then, I don't know, the 1990s, maybe 2000, hospitals put signs up everywhere, hand sanitizer everywhere, prevent the spread of disease, sanitize your hands. And so hospitals have gotten very good about educating the staff, the patients, and the visitors, preventing the spread of nosocomial infection. There are some other factors that you can look or read about how to prevent the spread of nosocomial infection, and you can see my online chapter notes to get further discussion of that. Any question about any of this? If not, let's move on to the topic of epidemiology. Epidemiology is the study of where and when disease occur and how diseases are transmitted in populations. The CDC collects and analyzes U.S. epidemiological information. Usually what happens is there are notifiable diseases that a clinician must report. They report it to their local health facility that reports it to their state health facility, and then the state health facility turns it in to the CDC. The CDC analyzes it and then publishes it weekly in the Morbidity and Mortality Weekly Report, which is usually called MMWR, and you can find the MMWR in the site www.cdc.gov. A notifiable disease are those diseases that physicians are required by law to report. Each state can have a different list of notifiable diseases, but most states have the same diseases. There's usually just a few exceptions. Any question about any of that? All right. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the total population in a given time period. Mortality is the deaths from a notifiable disease. The mortality rate is the number of deaths from a disease in relation to the population in a given time. Any question about any of that? Any question about chapter 14? All right, if not, let's move on to chapter 13. Let me save there. Chapter 13 is covering viruses, viroids, and prions. Here's a slide giving you the major goals and a rough outline. Always know the terms of the chapter. And if you want to get all the terms, you need to look at the objectives. These are only the major terms of the chapter. 2, understand the characteristics of viruses, their viral structures, and the classification of viruses. 3, understand about viral multiplication. Viruses reproduce in what we call a life cycle, and there are different life cycles, just a handful, but there are different life cycles, and we'll talk about them. And 4, understand about prions, what they are, what they cause, and what are viroids. Any question about any of that? The only goal of a virus is to replicate. How do viruses replicate? They infect a host cell and hijack the host cell and force the host cell to make copies of the virus. And that is how a virus replicates. Let's watch a little video on how viruses replicate. That's good.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions about that movie? Let me stop that. So let's talk about some of the general characteristics of viruses. When a virus is not within a host cell, it is generally inert. It displays little or no metabolic activity outside of its host cell. Viruses require a living host cell in order to multiply. So we can say viruses are obligate intracellular parasites. They must have a host cell in order to multiply. They multiply within the living cell using the host cell's enzymes, their nucleic acids, amino acids, ATP, and other molecules of the host cell. Viruses have no ATP generating mechanism. When they need ATP, they have to get it from their host cell. Viruses do have a genome, and it is either DNA or RNA. It is never both. It is either DNA or RNA. And yes, the virus genome may be RNA. The genome is the centralmost molecule of a virus. So here's a virus that cut away this side of the virus so we can look inside the virus. And there's the genome of the virus. Surrounding the genome is a protein coat, shown here, which all viruses have. Some viruses are also enclosed by an envelope, which is a lipid bilayer surrounding the protein coat. Not all viruses have an envelope, only some. Any questions about any of that? The host range of a virus is the range of cells that the virus can infect and multiply in. The host range is determined by specific host attachment sites, like sites on the cell wall or the flagella, and certain cellular factors necessary for viral multiplication. Most viruses only infect the cells of one or a few species, and usually they only infect only a few cells of that species. Is that clear? So most viruses usually only infect a few cells of one or a few species. The molecule on the virus that binds to the molecule on the host cell, we call that a ligand or an adhesion. So that's the molecule on the virus that attaches to the molecule on the host cell. The molecule on the host cell is called a host cell receptor. So the adhesion binds to the host cell receptor. And then the virus can bind to this cell and then infect that cell. So the virus does have to bind to the cell before it can infect the cell. Any question about any of that? And that's why the host range is oftentimes very specific. Here's a term that I want you to know, and that is a bacterial virus, meaning a virus that infects a bacteria. It's called the bacteriophage or bacteriophage. And sometimes it's called phages or phages for short. I won't use the term phage. I'll use the term bacteriophage. Any question about any of that? An example of a virus which is very specific is HIV. It only infects a few cells of humans and then one species of chimpanzee. It does infect T cells. I think it infects macrophages. It infects brain cells. But most other cells, HIV will not infect. It cannot infect a skin cell. It cannot infect a muscle cell. It cannot infect a bone cell. And don't quote me on that because I'm not about 100% certain what cells HIV cannot infect, but I don't think it infects any of those. But it can infect a macrophage, it can infect a T cell. And I think it can infect brain cells too. It does not infect other species other than that one species of chimpanzee. And then most species of the human, it does not infect. Viruses, when we're talking about their relative size, this slide is trying to show you the relative size of different viruses. Here we have a red blood cell where you're not seeing most of it. There is E. coli, which is a little bit larger than your average bacteria. Not much, but a little bit. Actually, this way, it's not. That is the average size, but this way, it's a little bit longer than your average virus. The largest virus by diameter is megavirus, shown here at about 440 nanometers. Ebola virus is the longest virus. It doesn't have the biggest diameter, but it's the longest virus. And it's just a little short of 1,000 nanometers or 1 micrometer. Which means that the largest viruses are about the size of the smallest prokaryotic cell. And then viruses go down in size all the way down to something like MS2. Let me blow that up so I can see it better. Which is the smallest virus at about 24 nanometers in size. But most of you have never heard of MS2. But you have heard of poliovirus, and that's the smallest virus you've ever heard of. It's almost as small as MS2, just a little larger. Any question about any of that? So the important part is viruses range from about 200 to 440 nanometers in diameter, meaning the average virus is about 10 times smaller than your average prokaryote cell bacteria. But your largest virus is actually just a little tiny bit bigger than the smallest bacteria. Any question about any of that? All right, let's talk about virus structure. A virion is a single mature, complete infectious viral particle. When a virus is replicating in a cell and the new viruses are made, we call them the new virions. Viruses are composed of different molecules. One that's important is the nucleic acid or the genome of the virus. Viruses can either have their genome composed of DNA or RNA. And yes, cells can only have their genome composed of DNA. If the DNA is DNA can be double-stranded DNA, and that's normal in a cell. Our cell's DNA is normally double-stranded, but the virus can have single-strand DNA. And that is normal for our cells also. Whenever the DNA is replicating or being transcribed, the DNA is single-stranded. The viral genome can be double-stranded RNA, and that is unheard of in our cells. Our cells do not have double-stranded RNA, or at least their RNA. Any question about any of that? The viral genome can also be single-strand RNA. If the viral genome is single-strand RNA, it's important to discuss whether the RNA is a positive single-strand RNA or negative single-strand RNA. Positive single-strand RNA is where the RNA can be read by the ribosome, so it will code AUG. The negative single-strand RNA is the complement to the positive. The ribosome cannot read negative single-strand RNA. Any questions about any of that? All right. The viral genome can be linear or it can be circular depending on the species. Linear DNA or linear genome is the way eukaryotes have their genome. Circular genomes are the way prokaryotes have their genomes. And then finally, some viruses can have their genome in several segments, like influenza virus has their genome in several segments. That should be familiar to you because humans have their genome in several segments. We call each of the segments a chromosome, at least in humans. In viruses, that term can be used, but I don't use it because it doesn't have DNA in the protein, so chromosome isn't quite the correct term. All right, any questions about any of that? Any questions about anything? We're a little bit behind Tuesday, Thursday. All right, if there's no questions about anything, I'm going to log off, and I'll be in the lab for only 15 minutes. So if you want to be in the lab and ask questions, make sure you're there in the first 15 minutes. All right, bye.